This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 10. Hey everybody, I'm Vincent Racaniello, and with me is Professor Emeritus Dickson Despommier.
Hey Vince.
You're looking summery today.
I'm feeling summery. I was out in this, not quite summer, yep, it's still the fall, spring. But we've had weather in the east that has resembled the summertime. So I was out pursuing my hobby of trout fishing.
I heard. Was it good?
Yeah, I actually turned into a hunter-gatherer yesterday and captured a brook trout, which I then took inside and cooked up and ate.
Was there any possibility that this trout had a fish tapeworm?
A very good possibility, in fact. Could have. But not the kind that we've already talked about. They have their own species that live in their own gut tracts that we can't catch.
We cannot get them.
But I can make a connection here, Vince, as to what I did yesterday, because I just learned on Saturday when I received my issue of Science magazine that the hominids that migrated out of Africa, Homo sapiens that migrated out of Africa, actually interbred with Neanderthals.
Correct.
And the number of, the percentage of genome that we now possess varies from 1 to 4% among those that continued the migration and spread themselves throughout the world. But it turns out that the ones that stayed in Africa, they don't have any trace of Neanderthal genes. Isn't that interesting?
Mm-hmm. Yeah. And what do you make of that?
Well, I think that some of us decided to stay at home and others decided to have this wanderlust that just forced them out, and then two separate groups of humans developed.
Interesting.
But yet, there are no genetic barriers, and in fact, there's no genetic barrier between a human and a Neanderthal. So, interesting. I think the whole way around is very interesting. Our origins are becoming more complex and more interesting as we advance our knowledge.
You've said before that you think we wiped out the Neanderthals with our infections.
I do.
So this means that we encountered them.
Not just encountered them.
No, we mated with them.
We inseminated them.
So that means it was very easy for us to give them are infectious diseases.
That's correct. There's a wonderful film made by a French director. I guess we could get it up on the internet right now. It's called Quest for Fire. Mm-hmm. And this is a movie in which, and I highly recommend it. That's my pick of the week, by the way, Quest for Fire. It involves a troop of Neanderthals who had this skull, and in it were burning embers from a fire that they had obtained somehow and maintained in their cave, and now they were migrating out of their cave because they were forced from their habitat by some disaster that befell them. And they've got this skull with the embers for the next fire because they don't know how to make a fire. And the movie is called Quest for Fire. And these Neanderthals encounter humans. And the story is very, very rich with interesting speculative anthropological data, but the part that I love the best is the part where, Neanderthal genes and human genes are actually brought together. There is an actual scene in the movie that actually depicts that, and it's not X-rated, but it almost is, and it didn't know that we were actually related to Neanderthals at the time, but now it turns out that we are. So this movie turns out to be prophetic in many ways.
Interesting. Yeah, it's set 80,000 years ago.
Yeah, and the other interesting thing about it is that the director of the movie had to actually invent 2 different languages, one for the Neanderthals and one for the Homo sapiens. And it was fantastic. I enjoyed the movie very much all 3 times that I watched it, and I'd watch it again. It's a great late-night movie, so that's my other pick. So,
all right, so that's
my first pick is to read Science magazine.
Yeah,
all right,
where the original sequence data were
that's
presented.
Correct. So, and my suspicion, and everybody else's is too, that malaria played a huge role into where we lived It probably prevented us from living in some areas. But as we will see, there's a genetic polymorphism that's actually encouraged to exist because it confers some degree of resistance to the most dangerous form of malaria. And so we could live in balance with malarial parasites if we had this particular aberration. Okay, so where would you like us to pick up the story?
Last time,
as you will recall,
we spoke mainly about the history of malaria. The fellow in India, the British fellow who found it was transmitted by mosquitoes.
Sir Ronald Ross.
Some of the Italians who... By the way, you mentioned the yellow fever guy. He actually played some role in
Walter
malaria.
Reed.
He did something about transmission.
Okay.
It was before Ross, I think.
Okay.
But he did play role. I said, no, that's the yellow fever guy, but I was wrong. You know much better than I do about malaria.
Not necessarily, Vince, but...
So today, let's move beyond the history.
Yes.
Even though the history is wonderful, we need to learn about the biology.
What the heck is malaria?
What is malaria? Malaria, bad air. So
exactly. And
it's a protozoan parasite.
It is. It's an apicomplexan.
Apicomplexan, exactly.
It's happy. It's a happy complexan.
It had many forms, doesn't it? Like all of the parasites we've talked about so far.
Apicomplexa is a big group. It's a big group of
What
parasites.
else is in it besides malaria?
We've got toxoplasmosis, Toxoplasma gondii, which we will discuss in detail at another time, and we've got Kryptosporidium, and lots of related organisms that infect tremendous breadth and depth of hosts. Okay. So malaria turns out to be a first-class parasite which has multiple hosts. Okay, some parasites only have one host, right? We discussed some nematodes, right, that we can only catch. Like pinworm is the one I can think of most. Nice. No other animal in the kingdom can catch pinworm except us. Isn't that interesting? And yet malaria, it turns out to be a primary parasite of mosquitoes, and we are the reservoir. We're not even the reservoirs. We're the intermediate hosts for a parasite which evolved to take advantage of mosquitoes as their primary definitive host.
Why would they do that?
No, well, we can't answer why questions here, Vince. Nature doesn't work that way.
Because
what I can say to you,
vectors usually.
Yeah,
right?
You think of them as that way, but mosquitoes carry the sexual stages of this parasite. We produce the pre-sexual stages, which the mosquito then ingests, and we'll discuss this.
Are we necessary for the life cycle?
We are absolutely essential.
Right. Is there any pathogen that exists only in mosquitoes?
Sure.
Okay.
I'm convinced they have their own Wolbachias, their own virals.
No, yes, the Wolbachias may be viruses, but the viruses we know of are the ones that happen to go into people or animals.
No, but I think there are mosquito-specific viruses that we sort of dismiss as little invertebrate type things that we're not interested in, not the pathogens.
Well, there's certainly insect viruses, but
yeah, exactly.
Whether they are
the polyhedron virus, for instance, and that sort of thing.
Okay.
Well, at any rate, let's pick it up at the beginning, shall we? Now that we've got this rich history behind us, because the history of malaria was the discovery of not only the parasite but the vectors, the sexual stages, and then finally putting it all together. The last link in the most deadly form of malaria was discovered as late as 1948.
And what would that be?
I can't jump to that one because it's a surprise.
But remember, we did talk about the drugs, the early drugs.
Yes, we did. Quinine.
The quinine and the one from the tree.
That's
quinine.
The one. Quinine from cinchona. Then derivatives of quinine, chloroquine, and then later mefloquine. Right. And now all of the quinolines are derived from the same basic parent compound. So let's start with the life cycle, okay? Now, how do you begin this life cycle? How does any life cycle begin? It begins by the stage of the parasite that infects humans. All right, let's start because we're anthropocentric. So we can start by saying that a mosquito, a female, this is important, a female Anophelene mosquito only. Remember we discussed the two Culisine and Anophelene mosquitoes. And so Culisine mosquitoes can transmit malaria to birds, but not to people. And Anophelene mosquitoes can transmit malaria to primates and to other animals, other mammals. And we discussed the difference of the resting position of the female mosquitoes, so that if it's an Anophelene mosquito, she's resting at a 45-degree angle to the wall, you know that you're running a risk of catching malaria.
Leave the lights on.
Leave the lights on. If the culisine mosquitoes are there, then there's no worry about that. But of course, then you have to worry about that.
Other things.
You're never safe. You're never safe. So, okay. So we have a female Anophelene mosquito.
All right.
She's harboring the infectious stage for humans, which is called the Sporozoite.
All right. Now, I have in front of me this lovely parasite book. It's actually called Parasitic Diseases by Dickson, Despommier.
And others, too. Now, don't please mention the other guys' names, too, would you please?
Guads, Hotez, and Nersch.
Okay, good. Guads.
Yeah.
Robert Guads and I were classmates.
His student used to be a PhD student here.
His son.
His
son used to be a PhD student. Got his PhD at here. His father, that son's father, who's one of my best friends, Robert Guads, works at the NIH, but got his degree in medical entomology at the University of Notre Dame, and I was there getting my degree in microbiology.
Small world.
It is a very small world.
Anyway, I have it open to page 54.
Right.
It's called Plasmodium falciparum or Falciparum.
Either or is pronounced correctly.
And this is the life cycle?
That is the life cycle.
So here's the female mosquito with sporozoites in her. And these are the, what did you call them?
Infectious stage for humans. That is the infectious stage.
What is a sporozoite?
I'll tell you. Well, to know that, we have to go back into the mosquito. So let's avoid that conversation first.
Looks like the picture on your bathroom wall.
Yeah, that's right.
All right.
You know what? The smallest things will do you the most harm, I can tell you right now. So finally, when you get down to the viral particles, forget about it. Biggest tapeworm in the world is totally harmless for people. So let's talk about just the sporozoite stage. What is it? All right, it's a haploid organism. Interesting, right? Haploid, not diploid.
Okay.
So now you know that we can't be the definitive host for this parasite because the only thing that's going to take place in us are haploid reproductive cycles.
Is this just a gamete? No.
No, it's not even a gamete.
Our gametes are haploid.
Yeah. It's not even a gamete. It's the infectious stage for humans.
Can this multiply?
We're going to talk about that right now.
What does it look like, Dickson?
It looks like a little sliver of nothing. If you were to give this to a pathologist on a slide stained up with Giemsa or any other stain of their choice, they would probably miss it and consider it schmutz on the slide.
Yeah, look at there's a picture here.
It's tiny. It's really tiny.
It looks like very elongated cells with a little nucleus in the middle.
That's what it is. It looks like a little crescent-shaped thing. All right.
And can you see this in a light microscope?
Of course, that is. I took that picture. So this is a light microscope picture of that.
Is it stained?
It is.
And where in the mosquito would you find sporozoites?
It's in the salivary glands and in the hypostyle. And the hypostyle is the part of the mosquito that she injects into us and then takes her blood meal.
Is it in her stomach also?
We'll talk about this.
Okay.
We'll talk about this. It's all over the place.
Right.
But the only places that it can do us harm are in her salivary glands and in the canals leading down into the hypostyle and in the hypostyle itself. Okay. So a female mosquito.
And she's fine having this sporozoid in her.
Well, yes and no. If she's heavily infected, there has been some speculation that heavy malarial parasite infestations in people can actually result in the death of mosquitoes and therefore act as a certain biological control agent to prevent overabundance of an outbreak. But
amazing.
It is amazing. And filaria works that way too, because filarial parasites infect black flies, and if they get too many, they die too.
Wow.
So let's go back to this cycle, because the female Anophelene mosquito she's not hungry, by the way, so she's not feeding on your blood. She doesn't use it as food. She uses it as protein source for her egg production.
Right.
Okay? So oogenesis in the female Anophelene mosquito is totally dependent on the protein that she derives from your hemoglobin.
Does she eat anything else?
She eats, and the same thing that the male mosquitoes eat, she sucks plant juices. That's what she prefers. But when she goes to lay a batch of eggs, if she had only fed on plant juices, she couldn't make any.
Okay.
She has to get her protein source from us. So in doing so, she has to overcome a number of barriers. Now, I could spend a whole program, or Bob probably would do a much better job, Bob Quads would do a much better job in explaining, and maybe we'll have him on the show, to explain all of the agents that a female Anophelene mosquito has to inject into you in order for her to successfully take up blood. She has to keep it from clotting.
Right.
She has to prevent you from determining where she is. Otherwise, you'll slap her. So she has an anesthetic that she injects as well. She has a vasodilator, which allows the capillary to spread wide open, or precapillary, because that's where her hypostyle is located.
She goes right into the capillary?
She does.
Not just into the skin.
Oh, she goes to the blood, man.
How does she know where the capillary is?
This is a very good question, Vince. She has incredibly sensitive receptors for heat and for chemo attraction. She can detect gradients of CO2.
Right, we talked about that last time.
She can also detect gradients of heat.
So remember last time at night when the CO2 of the birds come down the trees
that's right,
into the grass, the mosquitoes wake up.
Yep.
Now, tell us how a mosquito would land on you. It lands on you from the gradient, and then wherever it lands, it starts to jab?
She probes around with her.
Oh, she does probe.
Oh, she's got palps. The palps are there for sensitivity.
But she can't probe for too long, otherwise.
Exactly right.
We'll get them, right?
Exactly. It's pretty quick, actually.
Unless we're sleeping.
No, so what do you think she's looking for? Probably a difference in heat.
Because that would tell her the capillaries there. Oh, it must be so micro, right?
Not to a mosquito.
Well, to us.
When you get down to that level, boy, things look big.
Wow, that's amazing.
It is amazing.
So
it's
she's
truly
looking
amazing.
for a little bit of heat, which would signify the presence of a capillary.
That's right.
Is there any place on our bodies that they preferred to bite? No.
Not, well, the exposed parts, how's that?
Well, we're pretty exposed.
No, but what they don't do is they don't attempt to go through your clothing. So that means that the heat gradient is disrupted.
Yeah.
Okay, so they don't detect that. So they go for the CO2 levels. And
sometimes I feel mosquitoes biting through my clothing. Whereas I'm just imagining it.
No. If you've been camping out, if they're desperate,
I see.
They'll just probe everywhere till they find you.
So if you're sleeping and you're covered up except for your head, they'll bite your face.
Forget it. Or if you're sleeping underneath a mosquito net, as we'll discuss much later, and your back is leaning up against it, the mosquitoes can drill right through and get you.
Smart guys. Gals, I should say.
Yeah,
Gals.
you should. Please, no chauvinism here.
Wow.
All right, so you've got the mosquito successfully obtaining a blood meal, right? And in doing so, while she injects all this stuff...
So she's got her... What is it called, the part she puts in?
Hypostyle.
Puts the hypostyle into a capillary, and she has to inject anticoagulant and
vasodilator
and anesthetic, so you would feel her, because when she bites, it hurts, right?
Yep.
And how long does it take for her to have a blood meal?
That's a good question. You'll have to ask Dr. Quads. Actually, if you turn to the medical entomology section in the book, you can find a picture of his thumb being attacked by a mosquito. It's a famous picture that's been broadcast all around the world.
Is it in the malaria chapter?
Yeah, it is, actually. That's right.
Picture of Quads' thumb.
Yep. We're turning. Ooh,
these are wonderful pictures here.
They're great.
Oh, a graph. It's the end of the chapter.
I guess not.
It's not here.
No, turn back this way.
I think you're thinking of the other book.
Turn back this way. No, come on, let's go backwards. Yep.
Oh, that's his thumb, huh? What's coming out her rear end there?
Ah, great question. The mosquito's gut tract is filled with diverticula. All right, so if you imagine a balloon with fingers on it, and as the balloon fills, the fingers start to fill up. Multiple, maybe 10 fingers, 5 on each side. That increases the surface area, and it also increases the digestive qualities. It's stoichiometrically, it aids in blood digestion very fast. And this fills up with the red cells, and they pack. And then all the fluid part is discarded.
Wow.
So what do we call that process?
Centrifugation?
No. There's a special term for
Yeah, there
this.
is. Centesis of some kind.
Plasmapheresis.
Plasmapheresis, exactly.
It's called,
yes.
This was the world's first plasmapheresis laboratory.
Because it's done for people all the time.
Why is she doing this?
She doesn't want the fluids. She wants the red blood cells.
Correct.
Right?
She wants as many as she can get.
That's where the protein that she needs is,
That's
correct?
Right. So she discards the serum part.
And that's what's coming out her rear.
That's what's
By the way, the caption here is Adult Anopheles taking a blood meal from one of the authors, RWG.
That's right.
That's great.
Yep. That's his thumb.
So you don't know how long it takes to take a blood
yes,
meal?
I think it takes less than 30 seconds.
Okay. Now, she does this and then flies off. She's done.
No, but she barely flies off. I mean, she's laden with blood.
She's not going to take another blood meal for a while.
She can't go very far. In fact, she can only go as far as your windowsill. That's where she came in. So one of the control measures here is to take residual DDT.
Mm-hmm.
I know that word is a bad word for most people, right? But it's all we've got. So take your choice between malaria or the insects becoming resistant to DDT, and then you've got all kinds of other insect pest problems for your crops. But if you judiciously apply a spray of residual DDT along the windowsills, it's a fantastic control measure because you can sleep with your windows open. The mosquitoes can come in and bite you. But then when they go out and land, they absorb the DDT and they die, so they don't lay their eggs, and therefore the populations of mosquitoes are reduced, especially the infected mosquitoes.
But you've already been bitten, so...
Yeah, but you've already been bitten, that's true.
Yeah.
But it tends to keep down the number of mosquitoes that come into your house.
We're talking about in infested areas, not New Jersey.
That's correct. No, Jersey's noted for their mosquitoes, but not
Anophelene, though.
Oh, they have lots of Anophelene mosquitoes. The mythology about this disease is prescient. I mean, people all over the world are convinced that, if you don't have the vector, you don't have malaria. Well,
we have the vector.
We've never gotten rid of any vectors.
So we just don't have malaria here.
That's right.
All right.
That's right. So, okay, back to the story.
So she's taken her blood meal.
And at the same time, she's injected all these products from her salivary glands, including a hitchhiker. In this case, it's the Sporozoite Stage.
Along with all those things she needs to inject.
That's right.
So I'm looking at a picture of a mosquito here, which looks like it's mostly gut tract.
Well, that's because it's done schematically.
And then these are the salivary glands.
Vince, you're jumping ahead of the story here.
Oh, I am?
You're into the mosquito cycle. We were talking about the human cycle. It's on the page.
Well, I just wanted to know when she drank this blood where it went. So it goes into the gut.
Into the diverticula.
All right.
And then she digests it.
So when she bit you, she put in this hitchhiker.
That's right.
So
now what happens to the hitchhiker, right? This is an intriguing story.
Goes to the universe.
It is in the circulation. It can go everywhere. But it doesn't...
That was a veiled reference to Hitchhiker's Guide to the Universe, which only you would know.
That's right. It's to the galaxy, by the way. We didn't get to the entire universe, but...
It was galaxy?
Yeah, I think so. Hitchhiker's Guide to the Galaxy. So...
enters your bloodstream.
Someone I know will answer us and say, no, it was the universe, but I think it was the galaxy.
And then where's the first thing the blood goes to?
That's right. What is the first thing the blood goes to?
She hits a capillary,
She
so
does.
it's arterial blood.
No, that's not arterial blood. It's just oxygenated.
It's in the
Arteriole
middle.
Is the next
It's in the middle of the
that's right.
vein in the artery.
That's correct. So it's neutral territory, basically.
Right?
She's getting partially deoxygenated blood. What does she care? She just wants the blood.
Yeah, right.
Now, the sporozoites, though, get swept into the veins.
Eventually, right?
Into the venules, the veins, back into the heart.
Heart, pumped out, liver.
How did you know that?
Big
Vince,
filter,
how did you know that?
Big filtering organ.
Yeah, but they could go everywhere else, too, though.
Well,
not all the blood goes to the liver.
Where does the blood come out of the heart? The aorta, right?
Of course.
It can go up to your brain.
Good.
Or it could go down to your lower bits.
That's right. And by the way, most of the, in fact, almost all the venous return goes to the heart.
Mm-hmm.
Almost none of it goes to the liver, except from the mesenteric venules. That goes to the liver. So if she had bit you in the intestinal tract, they would all go to the liver.
But eventually everything goes through the liver.
Eventually.
And it's a great filtration organ.
It's a marvelous filtration organ, except in this case.
Well, it filters these guys, right?
It doesn't exactly filter them.
Well, the same with hepatitis viruses.
Yeah, I know, but there's a receptor involved in this.
Okay.
Sporozoite has a receptor on its surface, and it's called the circumsporozoite antigen. It's a quadripeptide, and I forget the sequence, but it's
4 proteins?
No, 4 it's 4 amino acids per peptide, and it's a repeated peptide.
Okay.
It's called a circumsporozoite antigen because it's antigenic also.
Mm-hmm.
And I believe that's the receptor molecule for the endothelial cells, which line the sinusoids
in the liver.
Of course. And the parasite attaches to that, all right? And then somehow it's carried inside to the parenchyma.
We don't know how that happens.
We don't.
If it were a virus, I could tell you.
It might be by diapedesis. It might be by some active process of the sporozoite itself. But the next thing that happens is that one sporozoite affects a parenchymal cell, a liver cell, an actual liver cell.
Hepatocyte.
An hepatocyte.
And when you get a bite of a mosquito, how many sporozoites do you get?
Varies. We'll
Depending
come back
on
to
how
that.
infested they are.
Correct. That's correct. Some are
Is
only,
one enough to cause malaria?
well, that's a good question. The experiments probably are very difficult to do because you've got to use them on humans. But I think in mice, I don't know how many they've gotten it down to in terms of the minimum dosage. I think it's way above 10, something like this, maybe 100. Maybe it's even more than that. With other parasites, I can tell you better, and you can with viruses too. There are minimum doses below which, if you don't exceed that, you don't get infected, even though it's viable organisms, right? Because your immune system knocks them out first. So what happens to the sporozoite is it goes into the parenchymal cell. Now let's, malaria is such a complicated organism. It's not just Plasmodium falciparum.
Sure.
We forgot to tell you how many different kinds there are before we jumped into this life cycle.
Well, we are talking about Falciparum.
I know that, but that's the most dangerous, but it's not necessarily the most interesting.
All right. What else do we have?
We have Plasmodium vivax, the lively parasite, which that's what it means. We have malaria, Plasmodium malariae, and Plasmodium ovale. And then we have
Malaria was the first discovered, right?
Just about. And then we have some related primate malarias that humans can catch occasionally. Plasmodium knowlesi is another one like that. And in Aeotis monkeys, this is a very important parasite. And so they can use Aeotis monkeys to look at a primate malaria that human beings could also catch. If you want them to really become infected, you splenectomize them. And then you can give them Plasmodium falciparum. But of course, then they all die. So it's a very short-term experiment.
So
those four are the major
human malaria.
They're called human malarias because they only infect humans.
And they can make you sick.
And they all make you sick.
They're all geographically distributed about the same, or they have
No.
specific...
They have restrictions, and particularly ovale and vivax. Wherever you find ovale, you rarely find vivax. Where you find vivax, you rarely find ovale. So there is another set of receptors on the red cell that we can discuss later, which relates to that.
And these are all transmitted by Anophelene.
All of them. Every single one of these species of Plasmodia are transmitted by Anophelene mosquitoes.
So let's see, the malaria in sub-Saharan Africa. Which one?
Two majors, Plasmodium vivax and Plasmodium falciparum. Today, most of the malaria in the world is Plasmodium falciparum.
And in Southeast Asia, same thing?
Same thing.
Okay. So the cycle...
But in West Africa, you have Plasmodium falciparum, Plasmodium ovale, but not Plasmodium vivax mostly.
South America?
South America, mixtures.
All right, so the cycle we started on was Falciparum.
Some people can have two species in the same person, by the way.
Really?
Yeah. Unlucky.
Delivered by the same mosquito?
Not necessarily. You can catch this by transfusion also. We'll get to that too. There's lots of ways we can give this. Drug addiction using a shared needle, this sort of thing.
Okay.
Tons of different ways.
So we're talking about
but
Plasmodium.
Not
transplantation.
Not transplacental. Not transplacental.
Okay, it doesn't cross the placenta.
Only in extremely rare cases.
All right. The cycle we began...
We tend to divert from the theme, so let's recap. An infected Anophelene mosquito bites a naive
individual. Infected with what? Plasmodium falciparum?
Falciparum. The sporozoites are injected during her taking of a blood meal. Notice I said taking a blood meal rather than feeding on us, because most people say that, but they're wrong. And then the sporozoite is distributed throughout the circulation, but because there's a specific receptor in the endothelial cells of the sinusoids of the liver, they stick.
And that's the only cell they stick to, right?
The only cell they stick to.
Okay. And then it gets into the parenchyma.
Right. It somehow is transported across the endothelial cells to the parenchymal cells, and then they enter the parenchymal cells. Now, with Plasmodium falciparum, there are two things that can happen. Let's talk about one first, the simplest thing. The sporozoite enters the hepatocyte and begins to replicate.
So it actually goes into the cytoplasm?
Into the cytoplasm and commandeers the host cell energy machinery.
So this is smaller than a hepatocyte, obviously.
Much
smaller.
Much.
Yeah.
No, that's the wrong picture.
I know, but
this is the picture.
Okay.
And it develops something called a cryptozoite.
It turns into a
Crypto
cryptozoite?
means you can't see them. Okay? And the reason why you can't see them is because they're not in the blood. They're sitting in the hepatocytes dividing. And boy, do they divide. You can get thousands of cryptozoites per hepatocyte
So
from
the
a single sporozoite.
Turns into cryptozoites.
Correct.
And they're all haploid?
Every one of them.
Morphologically?
They're little rounded things with a nucleus. They don't look like anything.
Okay.
When you look at them under the electron microscope, they're totally nondescript.
And the cell cytoplasm can be full of these, correct?
It is full of them. And then one day, it bursts, releasing the cryptozoites, which instantly are now renamed merozoites.
Even though they're the same thing.
Even though they might not be. You know what, Vince? They might pick up something from the serum that makes them different. But now know that each merozoite can never go back to another parenchymal cell. Once they're out of the hepatocytes, they can't go back.
So they reach the blood this way when the hepatocyte bursts.
Correct.
And what do you feel when that happens?
Nothing.
You're fine so far?
Absolutely.
No fever? Nothing?
It's called prepatent. You're absolutely spectacularly... It varies with the species.
Falciparum?
About 9 to 12 days.
And the burst.
And then it bursts open. Still nothing. It has to undergo some replication in the red cells next in order for you to start feeling sick. Because you can... Come on, you drink a beer and you can destroy more hepatocytes than this thing does. So the point is that the hepatocyte also has another stage of Plasmodium falciparum. It can just lie quiet. It enters the hepatocyte and just sits there.
This sporozoite sits there. Does it become a cryptozoite?
No, it becomes something else. Oh, and Vince's face just changed expression to say, oh, another surprise. Yes, this is a big surprise because we didn't know it existed, but it does explain a lot. Now, the listeners here will instantly recognize a mistake that I just made. Because what I'm really describing is not the life cycle of Plasmodium falciparum at all. It is the life cycle of Plasmodium vivax and Plasmodium ovale. So, for all of you listeners out there, just know that my mind was thinking, I want to tell them about this next stage. And I made a mistake, but I corrected it, and I said, nope, I can do this in two ways. I can either admit that I made some stupid mistake, which I did, or I can just transform it into another species, which I don't want to do. So what I'm going to tell you is, so the difference between Plasmodium falciparum and Plasmodium vivax is that when the sporozoite of Plasmodium falciparum enters the hepatocyte, it only has one choice, and that is to go ahead and divide into cryptozoites, which then transform into merozoites. And then you have to say, well, what do merozoites do? And the answer is merozoites infect red blood cells.
So they go into the blood, circulate,
yes,
encounter red blood cells.
And there are receptors which allow these merozoites to actually stick to the surface of a red cell, at which point the parasite induces the red cell to generate more membrane, which is remarkable because this is an anucleated cell. So it should be metabolically at its limit of whatever it can do. But, you know, there are some reserve membrane proteins floating around the cytoplasm of red blood cells. Why? We don't know. But the parasite takes advantage of it and actually causes the red cell to invaginate and surround the parasite, surrounding it with what they call now a parasitophorous vacuole. Now, let's go back to Vivax as a contrast, because I want to tell you about this. Because my friend, Bob Quads, played a major role in finding out the answer to this next question. And that is, and he didn't do it in 1948, the fact that malaria could relapse after you treat them triggered this research. And the research then culminated in a discovery. And the discovery was that some species of malaria use the hepatocyte as a reserve. They don't go ahead and replicate like a virus would. In fact, this resembles a latent viral infection. So it just sits there and waits and waits and waits. And we don't even know what it's waiting for, because with Plasmodium vivax, it could be there for up to 5 years.
No symptoms.
Nothing.
And it's in this latent stage.
And then every now and then, one of them decides to go on and replicate, and then you get a burst of malarial parasites, and you get sick again. They said, "Have you left the country? Have you?" No, I stayed at home. And the answer to that, of course, is they've got these Hypnozoite stages.
Hypnozoite.
Hypnozoite. Isn't that a great name for it?
Yeah. Is that Guadz's name?
Yeah. Well, there was a team that actually worked on this that worked it all out, but it was fantastic research that actually resulted in the discovery.
What's the form of a hypnozoite?
Well, the hypnozoite, if you use PCR to actually make it fluoresce, okay, you can find them in this huge bunch of liver, but otherwise it's very difficult to see them.
Because on this life cycle image of your book here, now we're on the Vivax page 56, they have a rupturing liver cell, and then next to it they have hippozoite, and there's a little red dot in the cytoplasm.
That's right, there is a little red dot in the cytoplasm. That little red dot is the reserve for Plasmodium vivax.
What is it? It's another form.
It's a sleeping sporozoite that's rounded up and formed one cryptozoite. It's a single cryptozoite.
I see. So this one above it that says cryptozoites, there are many.
There's zillions of them.
They make one big red mass. I got it.
So this thing it just rounds up and just sits there.
How does it do that, Dickson?
We don't know, and nor do we know what the trigger is for it to go on to develop into more cryptozoites by reproduction. But the fact is that we now know about it, and we can at least explain it. This is why no drug works against that stage, except one.
For Vivax.
That's right, except one.
But Falciparum, this doesn't exist, so it's not a big an issue. So what's the drug that works against it?
Primaquine. It's called primaquine.
All right.
And if you give primaquine to a Plasmodium vivax victim, and they can tolerate it because there are some caveats about the drugs.
It's toxic?
Oh, well, only for some people. If they have glucose 6-phosphate dehydrogenase deficiency, we'll get to that when we talk about treatments and stuff. But right now we're just talking about the life cycle. So let's go back now to the liver. The liver has these cryptozoites. Now they all burst open. Now we are going to call them merozoites. The merozoite, each one has the capacity to infect a red blood cell. Now, which red blood cells? You say, what do you mean, which red blood cells? There's only one kind, isn't there? Actually, there are several kinds. There are baby reticulocytes. There are normal red cells, and there are old red cells that are about to get processed by the spleen back into proteins, right? Some species of
we
mice.
have someone walking
We've got
in the ceiling.
mice in our ceiling here, folks. So I hope
you hear
don't
funny
mind.
noises. There are
Actually,
workers in the
more
ceiling.
like rats. So back to the story. I lost my train of thought.
It's their fault.
It is their fault.
So we have three kinds of red blood cells.
Oh yeah.
Baby.
That's right. We
red
have
blood cells and old ones.
The newly produced are reticulocytes.
They have nuclei, right?
They don't have nuclei, but they have ribosomes inside their cytoplasm, which you can actually see. So they're not finished making all their hemoglobin yet. They're needed so badly that they're thrown out into the bloodstream, minus their nucleus, but still in the process of metabolism. A normal red cell is finished synthesizing virtually everything it needs, at least we thought, but the parasite can force it to make more membrane.
Right.
And then the older red cells are sequestered by the spleen and recycled as proteins and stuff like this, or amino acids, I should say. So some of these parasites, like say Plasmodium vivax, prefer younger red cells. They will attack all the red cells, but if they're given a choice, they'll stick to the reticulocytes much more avidly than they will to the others. But Plasmodium falciparum appears to have no barrier.
Okay. Interesting.
So they go into the red cell, and now they're considered an intracellular parasite.
So they hit a receptor and they're taken in somehow.
Right.
And there's a nice picture here.
Right. So they live as an intracellular parasite in the liver. They live as an intracellular parasite in the red cell. And they live as a free-living parasite in the mosquito. Isn't that interesting?
As a sporozoite.
Yeah,
right.
that's very
now,
interesting.
so far we still have all haploid genomes.
We do.
Mm-hmm.
So the parasite now can undergo a replicative cycle in the red blood cells.
So now, is it in a vesicle of some kind in the red blood cell?
It's called the parasitophorus
vacuole.
We'll come back to that when we talk about toxoplasma also.
And it's basically separated from the cytoplasm of the red blood cell.
Completely.
Right?
But by a membrane produced by the red cell.
All right.
So now the parasite, what's it going to do? Let me think. Oh, yeah, I guess I should replicate. So it starts to divide, right? But to do that, it has to transform itself through morphological stages. And you said, oh, these are beautiful pictures when you got to the plates. The plates that we have shared with our readers are drawings made from microscope photomicrographs of the replicative cycle of all of the 4 species of malaria, and they're beautifully done.
In the red blood cell.
Inside the red blood cell. So each one of these stages has an ancient, when I say ancient, I mean back in the 1910s and 20s and 30s when the morphology was being described. These names were applied to those stages as a way of distinguishing them apart.
There are 30 different ones.
Well, it's a continuum, Vince. What you're looking at here is still slides from a movie.
Amazing.
So as the red cell that are infected to begin with, they all look alike. All 4 species look the same to the microscopist. And it's called the signet ring stage. And you'll see.
Number 6 here.
The early stages here of all 4 species look like a little ring with a little jewel at the end, a little pearl, a signet ring. So it's called the signet ring stage. And as it grows and begins to increase its size, it uses something from the host. What do you suppose that malaria parasites eat? What's their food?
Hemoglobin?
Correct. Well, you're half right.
Globin?
Right. And the heme part.
Do they want the heme part as well? They don't want that, iron.
That's a poison.
Yeah, just a globin.
For the parasite, it's a poison. We're going to come back to that because there's a drug that actually allows that to accumulate in the parasite and kill it. It's called chloroquine. Chloroquine's mode of action, we think, is to prevent the parasite from sequestering heme and getting rid of it. And so it dies in its own waste products, basically. So what you're looking at here is a parasite that, first of all, grows as a single cell until it gets to be a certain size. And then at a moment, it undergoes nuclear division. But it's interesting, Vince, the nuclei all divide within the same common cytoplasm.
And when is the nuclear division occurring? Like around 20 here in Vivax, I think?
Yep. When you can see 2 red dots,
stained,
when you can see 2 red dots, the Gametocyte stains RNA and it stains DNA differently, so you can see the difference between them.
So up until that point, there's this massive...
It's a single cell.
There's a massive kind of tissue-like...
That's correct.
What is that?
It's generating cytoplasm.
Cytoplasm, okay.
And why is it doing that? Because it's going to fill it with nuclei.
I see. Interesting strategy.
Very interesting. And then something later happens, too. Now, by the way, it's still accumulating heme, right? And it does so by a very interesting enzyme.
So it's accumulating it in the cytoplasm.
Yes, but it has a special vacuole for it. It's a waste vacuole. And there's an enzyme which allows this to happen. You know, heme is planar, all right? If you look at the heme molecule, the protoporphyrin 9 molecule, which, by the way, a Columbia professor almost got the Nobel Prize for learning how it was synthesized.
Really?
Yeah, and if I could think of his name, I'd tell you. I actually took a course from him a long, long time ago, and I can't remember. Oh, I wish I could come up with his name right now, because it was a very exciting set of lectures. It was in the very early days of using radioisotopes. And he was able to use labeled acetate to show how protoporphyrin 9 was actually made.
It wasn't Otto Funk.
No. He worked with Erwin Chargaff. It was way back in the days. Oh, I can't remember his name. Believe me, tonight at 3 o'clock in the morning, I'll wake up and tell you. I'll come up with his name. It doesn't matter. No, but the point is that hemoglobin metabolism is very important for us, but it's also very important for the mosquito because the globin part is really what they want. They use cathepsins. All right, these are acidic proteases. They use that to digest the hemoglobin into 2 parts. Dan Goldberg's work out at the WashU in St. Louis has done a lot of marvelous work on the isolation and characterization of these cathepsins. And so that's how we know it feeds. And it also sequesters the heme part, which tends to crystallize. Once 2 heme molecules find each other, they tend to auto-crystallize, right? Or they do it by themselves. So you could actually do X-ray crystallography on their byproduct, there's a term for it. It's called hemozoin. It's called that because it's not pure heme. There's a little peptide left where the enzyme cleaves it and leaves. Okay, I don't have to use that part of it. And this thing just tends to stack up. Okay, and the enzyme that causes this to occur is called a stacking enzyme. So it all seems to make sense, right? Okay, so as the parasite now divides its nuclei into this common cytoplasm, and in the middle of it, of course, is the reservoir for the waste products that it's generating. And as the red cell hemoglobin level drops, until finally it's mostly all gone, the parasite is now ready to break out and go on and infect more red cells, but it can't do that until it divides its cytoplasm also. And then at the last moment before it ruptures from the red cell, it divides its cytoplasm into individualized merozoites.
Yeah, now you have many per cell.
That's right, and each species produces a different number.
How many initially when the liver cell ruptures, when you have the first merozoites, how many red blood cells would be infected by those? A few? Many?
I mean, it could be 1,000. I mean, with each of these Kryptozoite stages that come out from the liver, you might find 1,000 different red cells infected all at once.
Now, in your picture here, they show the merozoites. Now, first they show a signet ring, which is the early stage, and then it replicates. It's called the cytoplasm. It's called the trophozoite. That's another.
Yeah, it transforms to the trophozoite stage. I'm putting that in quotes.
And the schizont.
The schizont is a Greek word for divide.
Okay.
So it divides. And like schizophrenia. It's a half and half, right? So this is schizont.
And then it looks like it becomes a merozoite.
Some call it a schizont.
Schizont.
Yeah. And then as the cytoplasm divides off, equally sequestering a nucleus inside, and by the way, it pinches off the toxic waste dump of hemozoin and leaves it behind. Isn't that a clever strategy?
Very clever. It doesn't want it, sure.
So it just pinches it off and says, you can have that little part, the naughty bits, and I'll get out of here.
And then it bursts out of the red blood cell?
It bursts out of the red cell.
And that's when you feel sick?
Well, eventually you will, because not right away, okay? At 9 days or 14 days when this pre-patent period starts, after the parasite has been injected, and then it undergoes its replicative cycle in the liver. Now it's about 9 to 12 days later, it gets out into the blood. Now, you count that as day 1. Now, how many days after that do you think without treatment you're going to have to go before you feel sick? You do the math.
At least a week, probably, no?
Well, make it a little bit longer, but not much longer. Not much longer.
7,
so when the doctor
days.
asks you, where have you been over your last 10 days or 12 days, you better remember. Because, within about maybe 20 days after you encounter the initial bite of the mosquito, you'll probably start to feel sick.
So these are merozoites that are released from the red blood cell.
Yes.
Very similar to the ones that left the liver initially.
Correct.
Same.
In fact, they're indistinguishable.
Right. And then they go on and infect more red blood cells.
Yeah, but they can't go back to the liver.
They don't
just
go back
remember
to the
that
liver.
They don't go back
well,
to
you
the
have
liver.
to be a sporozoite to get in the liver.
Correct.
So these guys just keep infecting. They're kicking around your blood, finding red blood cells, making more.
You got it.
Until you're
and
full
the body,
of these guys.
Eventually, the replication cycle could be very big very soon.
Yeah,
okay,
sure.
So when you get to a certain point, like about 1% of all your red cells are infected, that's when you start to feel sick.
What do you feel?
An initial infection with malaria, I've never had one, but I can tell you what people have told us as a teaching group and what I've learned from physicians that have traveled to these areas and experienced this firsthand, is that the first sign and symptom are not what you think. A headache and a backache. A headache and a backache, followed by fever.
Why the backache?
Good question. It almost sounds like flu-like syndrome.
Yeah, these are probably a consequence of inflammatory responses of the host.
Also, the release of the byproducts from the metabolism of the parasite. When the cells break, of course, out come this toxic waste dump stuff also.
Do these parasites stimulate innate immunity? I guess
We'll get to that. It's not innate immunity. They stimulate an acquired immunity because they can go unchecked and kill people. We discussed how much malaria there is in the world. There might be 2 billion people right now infected with malaria. And of those 2 billion people, less than 1% will die from this. But that's an enormous number.
But if untreated, a lot of people would survive.
Of course.
And they would.
Otherwise, you wouldn't be here.
What would happen? Where these parasites would keep multiplying in them?
No.
Self-limiting?
The immune system comes down pretty hard on this thing.
But if it's Vivax, you will have these hypnozoites. So
the immune system can't touch them because they're not active. But there's another question here that you should ask then. Well, what's the problem with malaria? Once you get it and you're immune, you shouldn't get it again.
Yeah, what's the problem, Dick?
But you get it again.
What is it, antigenic variation?
You got it. Bingo. Absolutely.
So you can be reinfected.
Yeah.
Just like influenza.
There's a waning of the immune responses from season to season. So it's a seasonal disease in many places. As a result, people suffer from it every single year. And as we'll see with this particular one, with Plasmodium falciparum, not Vivax, but falciparum, the most deadly form, there's a nuance to it. Let's go to that nuance now. Turn that page back. And you'll see that this particular parasite does not replicate in circulating blood. Now, you have to ask me some questions here, Vince.
I'm looking here at the photograph. It has the erythrocytic cycle.
Yep.
And it just shows some ring signet forms.
Yep.
But we don't see the multiplication of the merozoites as we did in Vivax.
Vince. So, by the way, the stages in the liver are referred to as exoerythrocytic stages, and the stages in the liver, of course, are referred to as the erythrocytic stages. So, in the capillary bed, or in this case, the pre-capillary bed, it's not in the capillaries, it's in the pre-capillaries. So they're a little bit wider than a capillary, okay? The parasite inside induces a receptor molecule on the outside of the red blood cell. But remember, it's inside of a vacuole, so how does that work?
Very interesting.
Right?
Yeah.
And it turns out that it's a histidine-rich protein. I mean 70% histidine. That's an enormous amount of histidine into a protein. And it sits on these little projections that the parasite induces in the red cell called knobs. And these little knobs interact with the endothelial cells. And it causes the red cell to become sticky. And it sticks to the precapillaries. And when that happens, of course, blood cells back up behind it. It slows them down. And what happens is then the oxygen level drops. Now, when the oxygen level drops, this parasite takes off like crazy. So it's a microaerophil.
Taking off mean multiplying.
I mean, they go crazy. They will not grow in an aerobic environment. So
this is different from Vivax.
It's much different from Vivax or malaria or ovale. So this parasite is a microaerophil, meaning that if you don't lower the oxygen tension in a culture medium, you don't get any growth.
So here in the red blood cells, they do not multiply until the oxygen tension drops.
Correct.
At which point then they will multiply.
That's
just
right.
as the Vivax forms do normally.
Now, you know what? We knew this forever, and we didn't cotton to how to grow this thing until someone got a clever idea. I knew both of these people, by the way, because they were at Rockefeller. One was named William Traeger. He was the most amazing man you ever want to meet because he was the perfect combination of a workaholic and a nice person. He was religious about coming into work. He would go into his lab, lock the doors, and conduct his research from 9 o'clock until noon, 3 hours. He would then leave instructions with his postdocs and graduate students. His door would open, he would come to lunch, and he would spend the rest of the day schmoozing science with the rest of us. He was fabulous. He was the best, and he was Curtis Patton, who was another good friend of mine, who we'll talk about him when we talk about trypanosomes. Curtis and I shared a desk at Rockefeller as postdocs, and we both agreed that we had never met an individual who was a better listener and absorber of knowledge than William Traeger.
He was trying to grow...
He was trying to grow malaria.
Which one?
Falciparum, of course, the most dangerous form, so you want to grow that one, right? So he tried everything. He had chemostats, he had pumps, he had oxygenators for the blood, he had mixers for the circulation. He had everything replicated except one thing, and that was the fact that somehow everyone knew, and he did too, that this parasite stuck to the capillaries and did something. So in walks this young postdoc Jim Jensen. Jim Jensen has become a good friend of mine. Taught at Brigham Young University. In fact, when he moved to New York to take his postdoctoral position at Rockefeller, he became the Mormon Bishop of Manhattan.
Wow.
In addition to becoming Dr. Traeger's postdoc. So, while he was in Dr. Traeger's laboratory, and we all called him Dr. Traeger. No one called him Bill. He said, "Come on, call me Bill." I said, "I don't think so." That would be like calling Barack Obama Barack. You call him President Obama, right? So we were in the presence of greatness, and we didn't want to cross that barrier because we just felt that he was such a wonderful man at that level. So, at any rate, Jim one day said to Dr. Traeger, "Have you ever tried growing these in a candle jar?" Well, the microbiology listeners out there know what a candle jar is, right? Because it's a big, thick-walled vessel you seal it with grease. It's got a top that you can seal with grease too, and the top rotates with a little plug at the end. It's actually a valve, and you can hook up a hose to it, and you can actually evacuate this if you wanted to, okay? Or if you want to get rid of all the oxygen, you can take a candle, put it inside the jar next to your culture, close the lid, seal it, turn the knob so that the little valve doesn't oppose the hole to go inside the vessel. So you've sealed it off, and just allow the candle to use up the oxygen. When it uses up enough oxygen, it goes out. By the way, that was Lavoisier's first discovery of what oxygen was, right? So here's this, we can relate this right back to the beginning of science. So you've got this candle jar, you've got malaria parasites in a culture dish filled with red cells. You've got the parasites inoculated from a victim, in this case an Aeotis monkey that's been splomectomized. Very expensive experiments. And now you're asking the question, will you grow in a reduced oxygen atmosphere? The first time that was ever asked was when Jim Jensen went to Rockefeller. They came back the next day, and they thought they had some evidence. They came back the next day, and boy, did they have the evidence.
How would you look? Just look at them under the microscope?
Yeah, you do how many red cells were there? You know how many infected red cells you put in, and then you can see, just like a plaque assay for viruses, you can just ask how many replicating cycles have gone on.
Had they tried growing Vivax before?
Other people had tried it before, and
Vivax
with
grew, right?
minimum success. But who cares about Vivax? It's the vaccine against Falciparum that you want, right? So that's the reason why they were concentrating on it.
So he got it to grow.
He got it to grow. You know what? Both of them were nominated for the Nobel Prize.
Never got it?
No, but they were at least nominated.
Yeah, that's good.
Come on, that's pretty damn good, I think.
I have to tell you, when my wife
I'd take that.
my wife went to Rockefeller to work on trypanosomes.
Right. She was with George, right? George Cross.
George Cross, and he was still there. They had taken away his lab, and he was just in the basement. And she said he was very sad.
I know he was.
Because they took away his fun.
They took away his fun. By that time, unfortunately, his wife had passed away also, so he lost his companion as well. So he lived into his 90s, though.
They used to go down and talk to him because they said he was a resource.
He's a marvelous person. Marvelous person. These are great memories, though, because, by association, you learn these things, and now you can share these stories.
So you were there when this happened?
No, I wasn't. I had left by that time.
Okay.
But I got to know Jim Jensen later on for another reason. But at any rate, so Falciparum malaria needs a reduced oxygen atmosphere in order to grow. That makes eminently good sense, because look at what it's doing in the capillaries, or precapillaries, I should say. All right. Now what happens?
Yeah, so these erythrocytes are full of more merozoites.
And more. And pretty soon you're going to die.
You feel really bad. Why are you going to die? Because you don't have enough red blood cells?
Something like that. Actually, in Falciparum malaria, there's another consequence of the infection that doesn't occur in the others. You become unconscious. In fact, it's known as cerebral malaria for that reason. And the way that happens is that this parasite induces tumor necrosis factor on the tissue side of the brain. This occurs all over the body, but in the brain, what would happen is tumor necrosis factor then induces nitric oxide. And nitric oxide actually puts you to sleep.
Right.
You can interfere with that process by putting in an amino acid substitute for arginine. And if you do that, it will not produce nitric oxide. And as a result, you can wake the people up. And Well, we get back to that too. It's called the Lazarus effect. So let's get back to the life cycle, though. Vince, it's a sexual parasite in the mosquito.
So I meant to ask.
Well,
these Merozoites,
how does this work?
These Merozoites are still haploid.
Yeah, everything is haploid up to this point.
And they're fine being haploid, right?
They're happy. Haploid.
So, all right, we got a person with a lot of Merozoites. They may be very ill, or sometimes they can recover.
Yep.
What's next?
Well, somehow you have to get this parasite from us, the intermediate host, to the mosquito.
Back to the mosquito?
So the mosquito has to come and bite us.
Mosquito who doesn't have any sporozoites?
Or infection, that's correct.
Now, it's just between us and the mosquitoes, right?
It is.
Nobody else involved?
Nope. So
why do we have to get a mosquito back to go through the cycle again?
It's the parasite's imperative, not ours. The parasite has to get back into the mosquito in order to make more diploid parasites first.
That's where the sporozoites came from.
That's exactly right.
So it just bites someone who's infected, pulls up some Merozoites, right? Is it simple as that?
Gone. Merozoites are digested. Forget about it.
Really?
Mosquito does not become infected.
Merozoites are digested because the mosquito just wants the globin.
No. We're drifting here. We haven't talked about another stage that the parasite produces. It's also haploid. It has nothing to do with its replication.
Okay.
This is exciting because, Vince, it means that this parasite differentiates into two different groups. A reproductive set of just dividing and conquering, and another pre-reproductive stage, which is sitting there waiting for a mosquito to take it up. Now, if we could prevent that stage from developing, we could prevent malaria.
Sounds like it, yeah.
However, it's a point of, once the Merozoite enters the red blood cell, it can take one of two paths. One is to go on and divide, and the other is to become one of these pre-sex stages called a gametocyte. That's why they call it a gametocyte.
Sure.
Okay, so you have two kinds of gametocytes, right?
A plus and a minus.
Male and female.
Yeah.
So the gametocytes of all four species are distinct morphologically, so you can recognize them by doing a blood smear, and just finding those stages, you can tell what kind it is.
These are free-living or inside the red blood cell?
They're inside the red blood cell.
So instead of replicating as Merozoites, they differentiate into
gametocytes. And we don't know what the triggers are. We don't understand this. We're looking at it, and we're getting ahead of the game because we've got the complete genomic sequence for Plasmodium falciparum in our possession. It's a very AT-rich genome. Very AT. It was very difficult to sequence.
So the parasite is making lots of Merozoites, but at the same time, it's also making gametocytes.
Correct.
And it's making two different kinds.
It is.
What, do they have a different name?
Yes. The macro gametocyte and the micro gametocyte.
And let me guess, the micro is the male.
Yes.
Are you serious?
I think so.
Okay. Why are they macro and micro? What's the basis for that? It's morphological?
It's based on morphology. It's not important.
But they're both haploid.
They are still both haploid.
And I think they're going to combine at some point and make a diploid.
They might.
They do that in the mosquito?
They do that.
So mosquito has to get both of them in a blood meal.
They do.
These are free-living now, not in erythrocytes.
No, they're in the erythrocytes.
All right, they don't burst out.
They don't. They are digested out by the mosquito.
All right, that's why you wouldn't want to eat a Merozoite, because, as you said, the mosquito would digest it. All right, so you eat a red blood cell, which the mosquito is going to protect.
No. The mosquito won't protect the red cells. The mosquito wants those red cells.
Yeah, it wants to eat it, but it brings it into its gut.
And then it digests everything.
And this guy gets out.
Right. It digests everything except... Everything else is digested, including the Merozoite stage of the mosquito, of the malaria parasites.
But the gametocytes
are resistant
to
insect digestive enzymes.
Excellent.
In fact, they're stimulated to go to the next differential stage.
By the digestive enzymes? Amazing. And what would that be?
Well, then they become the sex cells.
Mm-hmm.
Okay? And these two sex cells then mate. And it's interesting.
In the gut of the mosquito?
Yeah, this is the part of the life cycle that Laveran discovered. And it's because he was working on collecting malarial parasites from birds. And then on his bicycle, he was riding back to his laboratory in North Africa. And by the time he got back to his laboratory, these stages had actually developed in the test tube. Right? It actually deteriorated enough to allow this to occur inside the test tube. So you can get this to actually occur outside the mosquito too, but it goes in much less efficient way.
So just in the lumen this happens?
In the lumen of the intestine.
The cells fuse.
Yeah. And they form something called the ookinate. Ookinate.
It's not in your picture here.
No, we don't know what that thing looks like too much, so we don't even talk about it. But, remember, this book is written for medical students and clinicians. Why would they need to see a picture of that? So the ookinate is now a diploid organism. And what happens is that it actually squeezes in between the columnar cells of the stomach of the mosquito. And the mosquito's stomach is coated with a membrane. And this ookinate lies up underneath that membrane between the columnar cells and the hemolymph of the mosquito itself into the blood space of the mosquito. Okay, and it now undergoes reduction and division. And what do you think it becomes? It becomes something called an oocyst.
I was going to say sporozoite.
Well, inside the oocysts are the sporozoites. So, aha!
So it now becomes haploid again.
It is haploid again.
Sporozoites are all haploid.
So there's the story, Vince.
That's the whole cycle.
That's the entire cycle. Now, interestingly enough, you'd think that, well, in order to get the sporozoites out of these oocysts, somehow they would have to rupture and let them come out, but that's not what happens.
What happens?
The sporozoites actually swim out into the hemolymph. And the hemolymph of a mosquito, or any other insect for that matter, is a clear fluid, contains some lymphocytes or hemolymph cells, right? But mostly it's a clear fluid. And there are 4 open chambers to the heart of this organism. And it circulates the hemolymph and bathes all the organs. And in doing so, it brings the sporozoite stage from the gut tract, or the area surrounding the gut tract, to the salivary glands. When these sporozoites reach the salivary glands of the mosquito, they actually penetrate into the salivary gland. And they just keep accumulating inside, okay? So there must be receptors for that.
Yeah, I see.
Which is interesting because the sporozoite also has a receptor for the endothelial cells of the sinusoids of the liver. So they've got 2 different receptor molecules here, maybe more.
I was wrong. There is a mosquito cycle here.
Oh, well, of course there is.
It's called sporogony.
Sporogony.
It shows them ingesting the gametocytes.
That's right. That's the name for the process.
Gamete production.
That's right.
In the stomach.
And they call it exflagellation because what they thought they were looking at were flagellae, but what they're really looking at are sperm.
Yeah.
And each one of those can now inseminate or mate with, if you like, a female
gametocyte.
Microgametocyte.
Gametocyte, right.
Macro and micro.
And then it says oocyst formation in the wall of the stomach, as you said.
But of course, I worked with the artist pretty closely
and
on
then
this.
The release of the sporozoites in the stomach, and then they migrate to the salivary glands.
That's correct.
And then when they bite you, it starts all over again.
It starts again. So here we have it. We've got a haploid organism injected into humans. We accumulate lots of that stage, plus the pre-sex stages. The mosquito ingests blood with all of that, discards everything but the pre-sex stages, which then develop to the sexual stages, mate,
Correct.
Producing an ookinate, producing an oocyst, producing sporozoites, back to square one. Now, that's an ancient life cycle that's been going on for many, 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 many millennia. Probably before humans, way before humans, because mosquitoes arose some time ago. So to have the mosquito as the primary host tells you something about the evolution.
There had to have been another intermediate host before humans.
Sure, but you could pick anything.
Sure.
Amphibians, reptiles, anything.
All right, so I have many questions, but I think we should do them another time.
Fine.
I'd like to know about the disease, immune response,
malaria
and drugs.
Is so important that we can devote 5 sessions to this if you'd like, but I'm going to run out of information around session 3. So I'll have to call on some of my friends to help out here.
You could get someone else to help out.
Now, you're probably people listening to this will say, how does this guy know all this? Well, I just want to tell you what I was an emeritus professor of I'm an emeritus professor of parasitic diseases, and I've taught this course for 38 years here at Columbia. And every year, I want to teach the malaria, and I do it for a good reason, because I do it to keep myself abreast of what's going on. Robert Quads has been an enormous resource on this, but Dr. Charles Kanersh also. He conducts, drug trials and things like this, because he's partially employed, or I should say almost fully employed now, at Pfizer Inc. Mm-hmm. And they're involved in a huge program to help eradicate, or at least control, a lot of infections. And this is one of the ones that they're concerned about. So I get a lot of help from my friends, as they would say. And so I thank all of those people for contributing to my knowledge base and allowing me to convey this to a wider audience.
You stand on the shoulder of giants. Who said
That's
that?
Right. I believe it was Wayne Newton.
No.
It was
a scientist.
It was
Newton.
It was Newton, but it wasn't Wayne Newton.
If I do good things, it's because I'm standing on the shoulders of giants. We all do, Dickson, right?
Exactly. Well, I don't think I'm standing on the shoulders of giants right now, but I certainly did when I was at Rockefeller.
Well, everything you know is based on
what you hear and what you read.
What you read, other people, yeah.
Sure, of course.
I'll read a couple of emails here. Okay?
Please.
And we'll wrap it up.
Please.
First one is from Becky. I am a second-year graduate student, and the lab that I joined works on Leishmania and trypanosomes.
Cool stuff.
I originally discovered TWiP via TWiV, where my true interests are, but that's a long story. Matt Freeman came and gave a seminar and gave a small plug for TWiV. I am really excited to have the opportunity to listen to highly informed people talk about parasitism. I request more frequent episodes. I will start prepping for my qualification exams soon, and I am sure that this podcast will be invaluable, especially...
Don't blame me if you fail.
if you get to Leishmania soon, hint, hint. I would also like to say that listening to a podcast is like having a viral parasitic infection. Usually, there are only a few people that show clinical symptoms, i.e. send an email or post a comment. But there are actually many more who have asymptomatic infections, i.e. just listen. So in the end, there are many people who would like more episodes, but only a small percentage will actually contact you to request more. I'm very grateful for the time you both take to prepare and do this podcast. Keep them coming.
What a thoughtful letter. Thank you so much for your comments.
Bernhard writes, Dear Twivers and Twippers, last time I suggested a pick of the week, but now I'm ready to pose a question. In a Science News compilation, I recently learned about the work by Shigeto Yoshida et al. Of Jichi Medical University, supposedly reported recently in Insect Molecular Biology, on genetically engineering mosquitoes to secrete an antigen from Leishmania in their saliva. The idea being that being bitten by such a mosquito would trigger antibodies against the protozoans. The regulatory issues with such an approach are, however, quite severe, since you cannot control who gets the shot. However, I've since heard a podcast on Australian wildlife being threatened by picking up diseases from farm animals and species introduced by the Europeans, like rabies. Would it be possible to use the above approach to immunize wildlife, say, using a mosquito that's specific to the wild animal and doesn't bite humans? Would the supposed specificity help with the regulatory issues and provide a proof of concept for the approach? Just an idea. Hope you can keep TWiV and TWiP going for a long time.
Oh, we do too. Thanks for the comment. And I know there are some efforts involved in making mosquitoes refractory to malaria. Yeah. And releasing those mosquitoes has ethical issues surrounding it also, because you're really releasing a competent vector in lots of other ways. I mean, just because they can't transmit malaria doesn't mean they can't transmit filariasis or dengue or lots of other things. I wouldn't mean dengue, but things like that. So people will object, no matter what, to the release of mosquitoes, no matter how well-intentioned it is, because it's really releasing something that is a disease transmitter. Encephalitis viruses, for instance, right? So it's a tough call, I think. But it shows you how desperate we've become for solutions. That's the main thing, I think. When you examine these life cycles and you think, now that I understand everything. We've got the human genome sequenced. We've got the parasite's genome sequenced. And what I didn't mention was that we've got the mosquito genome sequenced. Somehow you should be able to work out some ways of controlling everything, because you know all the proteins that are encoded for. You know what? You just know the alphabet. You don't even know how the sentences are constructed. You don't know anything about paragraphs, pages of text, chapters in books, references. The whole thing has to be worked out, right? And we are not even close to that for ourselves. So I think having that data in your hands doesn't allow you to go to the next step that you'd like to go to, and that is to throw this into a computer and say, okay, show me how this works.
Would you be able to make a mosquito species specific so it didn't bite humans, but
here's the idea that I've been thinking about all my adult life since I've heard it's since I've heard it's necessary to do this. Unlike certain viral infections where the mosquitoes, when they become infected, can transmit this to their offspring. That's called vertical transmission. Malarial parasites cannot be transmitted to the offspring of malaria-carrying mosquitoes, which means that in order to effectively transmit malaria, a mosquito has to bite you twice. Not you, but it has to bite an infected individual first, acquire the infection, and then bite somebody else. Now, why can't you just genetically engineer mosquitoes to bite once? You know what? They'll be perfectly happy, they can make more mosquitoes, and we will never catch malaria. What's wrong with that idea? Excuse me. While I go back to my dream. So, I've had this conversation with Bob Gwadz because he's a mosquito geneticist by training, and he says so few of the population of mosquitoes actually bite twice, but that's enough to carry this infection. You'd think, how many times do you have to be bitten in order to get malaria? And the answer is about 100 times.
Really?
That's a lot of times, right?
Is that right? I didn't know that.
That's right. But we'll come to that when we discuss the vector part of this. But guess how many times you get bit if you're living in a malarial transmission zone per day as you're a little kid?
200 or 300 times.
Try 1,000.
1,000?
Yeah.
It's a lot.
It's almost like 100% sure they're going to catch malaria. It's a lot. But they're so desensitized to the mosquito
100
bites.
100 times a minute.
They don't even know if they're being bitten.
100 times an hour.
They don't even know they're being bitten. So the big solution to malaria right now, out of desperation, is bed nets impregnated with insecticides to try to lower the transmission cycle. And there's a guy here by the name of Jeffrey Sachs who's been a champion of that, and it's worked. He's been able to lower the transmission of malaria he hasn't been the only one, of course, but he's the one that's championing this, by about 50% in some areas. But in order to lower the death rate from this parasite, you have to do more than that.
So this makes people very unproductive, this illness, right?
Incredibly. You can get it seasonally, or you can get it intermittently throughout the year, or you can get it continuously. That's the different zones that are transmission areas. So when it debilitates a person, they're out for weeks. Right? Out for weeks.
I understand.
Then they recover. Then they go back to work. They're a little weaker than they were before. They gain their strength just in time for the next outbreak. It's maddening. Now, most adults don't die from the infection. It's just mostly children. So that's the biggest tragedy. Is you're just cutting the population out at their knees. It's just a horrible set of circumstances.
All right. Thanks, Bernhard, who's from Switzerland, by the way.
Okay.
Ryan writes, I love the podcast. I'm a grad student in microbiology, and this podcast, along with TWiP, is a great part of my week.
Great.
I study bacteria, so we need a TWiP.
We do. That's right, a TWiP.
Fungi.
That's right.
I could do one a week. TWiP, TWiP, and TWI. TWI being immunology, I guess.
Don't get us started.
Ashley writes, Dear Dr. Despommier, my name is Ashley, and I'm a graduate student at Columbia University and an enthusiastic fan of both TWiP and TWiV.
Cool.
She's actually working in my laboratory.
She can come and see me if she likes.
Recently, I was listening to the podcast This American Life, episode 404, entitled Enemy Camp, which features a segment called As the Worm Turns. This brief story was about a man who believes that hookworms have cured him of his allergies and asthma by acting as immune system modulators, in an example of what has been coined the hygiene hypothesis for immune system dysfunction.
Right.
The man subsequently went on to sell his hookworms to other individuals under the guise that they are a cure-all for autoimmune disorders until he was threatened with arrest and fled the country. She gives us a link to this episode, which we'll post. Considering that This American Life has such a large audience, I'd imagine that this story has generated a lot of interest in the hygiene hypothesis among the general public and would make a great topic for an upcoming TWiP episode. Having had an interest in autoimmune disorders and the hygiene hypothesis in particular for years, I think it would be great to hear your take on this as an expert in the field of parasitology.
It's a great letter. Look forward to meeting you. And the answer is that it not only... the mechanism that we think is at work here is not a modulatory effect, but rather a diversion effect. Here's the deal. As we evolved over time, and I think we just discussed this a little bit when I was discussing nematodes, your immune system was keyed into that, and it's mostly an IgE and an eosinophilic response that knocks out these parasites, these big ones. And other immune mechanisms too, but those are the two main bodies that come to bear on the worm to make life uncomfortable for the worm, kick it out of the intestinal tract, and away it goes. Along the way, its repertoire for response included some of the host tissue antigens that are normally present. And this is a subset of people that develop a disease known as Crohn's disease. Crohn's disease has as its base an antibody and cellular attack on your gut tract itself. And anyone with it will tell you it's the worst thing that could possibly happen to you because it's chronic, long-term, you've got it, and there's nothing you can do about it. Until recently, when they experimentally, out of desperation again, tried to restore the original targets to the host, which were worms, as it turns out. And infecting someone with Crohn's disease with a worm called Trichuris ovis, which is the parasite that infects sheep, the whipworm of sheep, infects some people, okay? And you can use other lookalike Trichuris analogs to infect other people with it. And in any case, the end result was that it modulated the immune response against your host tissues and refocused it on the worm. Worms were eventually kicked out, however. But during this 3 or 4 year period when the worms were still present, the symptoms from Crohn's disease went away. So this is called worm or helminth therapy. It's a ridiculous hypothesis, but it seems to be true that we evolved to take care of these parasites, and the moment we got rid of them from our environment by control measures, our immune system, in some of us, not all of us, just a few, said, I've got to take target practice, otherwise I'm going to get stale. And it finds your host tissue instead. So now, in order to get it to refocus again, and I think hookworms serve the same purpose in this case.
Interesting. Diversion, yeah.
Immune diversion, that's right.
Well, there's also a wider idea that you should have certain infections as a child to make you more educated later on. You should have more viral infections. Or
I will never catch anything if that's the case because I used to wallow in it.
So is there anyone who works on hookworms that we could have someday?
There is. Dr. Peter Hotez, another author on our book here, by the way, I must tell you. He's the world expert on the development of a vaccine against hookworms, but I'm sure he'd be glad to talk about this too.
Do you think he would join the podcast for an episode?
I know he would. I'll twist his arm.
All right. Thanks, Ashley. We have lots more, but I think we'll end this show at this point. We'll save them for next time.
Okay, to be continued in malaria is a 4-part series.
Yes.
Because we have the clinical aspects next, and then we have to cover global control.
Beautiful. I'd like to know about the immune response also. I noticed there was a
so would everybody else.
There's a graph in here.
Yeah, we can talk about it.
Vaccines, drugs.
The lack thereof. We
have a fellow here working on drugs.
We do.
Trying to understand why resistance.
Dr. David Fiddick, and I think he should be a guest on our show too.
So
he's terrific.
Which number do you think? Number 3 or 4? Okay, I'll work on them.
Number 3.
If you are a new listener to TWiP, please subscribe in iTunes or the Zune Marketplace. Helps you get all the new episodes automatically. And if you don't use either kind of aggregator, go over to microbeworld.org/TWiP, where all the episodes are stored. You can play them there, and we have some show notes and photograph as well. And if you like TWiP, tell your friends. And always great if you can write a review in iTunes. That helps us a lot. TWiP's also part of sciencepodcasters.org and promednetwork.com, which are websites where you can find other high-quality science podcasts. As always, send us your comments and questions, twip@twiv.tv. Good to see you again, Dickson.
Vince, it's a pleasure as usual.
Wonderful stuff.
Yeah.
I hear you're off to Mexico.
I am, for a little R&R.
Birthday boy.
There you go.
Happy birthday, Dickson.
Thank you, Vince.
And enjoy. When you get back, we'll see you again.
We will, and we'll pick up another TWiP.
Thanks, everyone, for listening. And what have you been listening to this week in parasitism? Thanks for joining us. We'll be back soon. Another TWiP is parasitic.